Welcome to this video about the Master of Genome Analytics at Monash University. I'm Professor Robert Bryson Richardson. I'm the course director, and alongside Dr. Hendrika Dovenvoorden, the course coordinator, we run the course. You'll be taught by a range of experts at different areas of the university, so this is a course taught jointly by the faculties of science and the Faculty of Medicine, Nursing and the Health Sciences, with expert contributions across those faculties and including our clinical schools and colleagues in the Faculty of Arts. They have a broad range of expertise from the fundamental science in genetics and genomics to the application of this technology in diagnostic settings. So you'll be taught by a range of experts, all focused on their specialist areas. I'd like to begin by introducing one of those, Dr. Desiree Desarte, who will tell you more about the course.
Hi, I'm Dr. Desiree Desarte, and I'm one of the lecturers in the Genome Analytics course. Let me tell you a bit about myself. I have greater than 25 years of experience in the field of genomics and genetics. I'm well connected through my roles in service delivery, research, and high-level committee roles that allows us to develop best practice standards and guidelines in the field of genomics. I have trained many scientists, trainee pathologists, and also students to become genome analysts. We are living in an exciting era. The powerful new genome approaches are transforming healthcare worldwide to become the new standard of care. Many of the developed countries have embraced this new technology to implement into their healthcare services. The Global Alliance of Genomics in Health has estimated that over 60 million patients will have their genome sequenced by 2025. This will generate a tsunami of complex data. So genome analysts are urgently needed to interpret all this new data to allow specialists and clinicians to provide personalised services to their patients and their families.
The Monash University Master of Genome Analytics has been specifically designed to give our students expert training in genetics, genomics and bioinformatics. The aim of the course is for our students to be job ready, and therefore we offer students exposure to a realm of concepts including inherited diseases, microbiome, cancer, infectious diseases and more. My role as the course coordinator is to help students feel like they are part of our course and also help them with any course inquiries throughout their progression. I also organise social and networking opportunities for our students, including careers nights where you can meet and network with professionals in the field. So why would you choose the Master of Genome Analytics? It is a comprehensive training for the next generation of diagnostic genome analysts. Without these specialist genome analysts, we cannot translate and expand the discoveries from genome sequencing to the healthcare system. And this means we'll ultimately be able to improve health in our society. So you will be part of a graduate cohort able to meet the growing demand for expertise in the analysis of genome sequence data. The Master of Genome Analytics is run here at Clayton campus at Monash University in Victoria, Australia. The course takes 2 years full-time, but can also be taken part-time, where it would take 4 years. For those students with a background in genetics and genomics, we offer a fast-track version of the course which takes 1.5 years to complete. So those students commence in February, whereas the students taking the full 2-year version of the course commence in July. The requirement for the course is a 60% or credit average in your prior studies. The delivery mode of the Master of Genome Analytics is blended learning, meaning we provide videos, content and information online for you to be able to go through at your own pace when it suits you. We also have face-to-face classes, so you can come in, meet with fellow students and of course the experts in the field, your educators, to go through workshops and practical laboratories to help you understand more closely what it's like being a genome analyst.
The course is structured in 4 parts. The first part, Part A, Genomic Foundation Studies, is where you'll start if you're coming from a previous study in biology, but without experience in genetics or genomics. And you'll get an introduction to cell biology, human genetics. We'll have a unit focused on the application of genetics to medical areas, and you'll learn about some of the biotechnology tools that are used in this discipline. If you've come from an undergraduate degree where you've covered genetics or genomics or a related discipline, you'll enter the course in parts B and C. These are taught simultaneously. There's the core studies in genomics, which is more focused on the fundamental concepts, where you'll learn about genomics and its applications. We have a unit focused on the sequencing technologies that underlie all of these approaches. You'll start to learn about bioinformatics, where you'll learn the necessary skills to analyze this genome sequence data, although I'll stress that no prior experience in programming or computational science is required. We also have a unit that focuses on examining the different sequences within the genome to help you understand what it means when they're mutated. Alongside this, you'll undertake specialist studies, where you begin to focus on the area of diagnostic genomics. We have a unit on genome curation and clinical applications of genomics, where you start to look at how to identify variants in disease samples, the standards that you have to apply to their analysis, and the reports that you need to prepare to report these findings to the clinical teams. In the final semester, you undertake Part D, and this is where you have a choice of pathways you can undertake. The first pathway is a research pathway, where if you're more interested in research or would like to use this as a pathway to a PhD, you can undertake a 24-point semester-long research project in the area of genomics. The other two pathways have some overlap. In both of them, you'll undertake 2 units, one focused on disease surveillance in the microbiome, where we look at the application of these technologies not to inherited diseases, but to infectious diseases and the microbial content of the gut. So this could involve looking at the recent strains of influenza or COVID, or the impact of the microbiome on health. Alongside that, you'll undertake a unit focused on cancer genomics. Again, similar technologies and approaches, but very different mutation types, and an understanding of these can help inform treatment. So alongside those 2 units, you can then either undertake an internship where you'll be embedded in a local diagnostic lab, and you'll do this for half of the week throughout the semester, or you'll undertake the advanced case studies unit, where you'll emulate the workplace and will introduce you to more advanced concepts in genetics and genomics in this application. So where will the Master of Genome Analytics take you? We designed the course very much focused on a diagnostic career outcome, and you might see this profession referred to as genome analyst, or genome curator, data analyst, or clinical variant analyst. These are all as different terms referring to the same profession, as part of the healthcare system, analysing data and reporting clinical variants of interest. It can also be used as a pathway to PhD. Whilst we set up this course with this career goal in mind, these skills are applicable to a range of other areas. So possible employers include diagnostic laboratories, but also of growing interest in biotechnology companies, pharmaceutical companies interested in exploring pharmacogenomic approaches, and of course in university and research institutes investigating genetics and genomics. I'd like to thank you for listening and point out a couple of places you can go for more information. You can look at our course website, available at the address shown here, or email us if you have further questions. Thank you for your time.